What about ILOs of this lecture? By the end of this lecture, you will be able to, number one, classify receptors according to their histological structure. Number two, you must describe structure and the correlated functions of the Pacinian corpuscle. Number three, you must describe also structure and the correlated function of the Meissner corpuscle. Number one, nerve endings. Nerve endings, they are structures that represent the site of contact between a non-nervous tissue and the terminal end of peripheral nerve fiber, which means the point of contact between non-nervous tissue and terminal end of peripheral nerve fiber. Receptors are subdivided into two types. The first type is called unencapsulated receptor, and the second type is called encapsulated receptor. Unencapsulated receptor, that means that this receptor is not covered by capsule. So, letter B, which is called encapsulated receptor, is covered by capsule, as this receptor is covered by connective tissue capsule. What about the types of unencapsulated receptor? They are subdivided into three types. The first type is called free nerve endings, second type is called peritracheal nerve endings, and the third one is called Merkel's tactile disc or Merkel's disc. As regards encapsulated receptors, they are subdivided into different types, several types. 7 types of encapsulated receptors from 1 to 7. The first one is called Meissner corpuscle. The second one is called Pacinian corpuscle. The third one is called Raffini corpuscle. The fourth one is called Krause's end bulb. And the fifth one is called muscle spindle. 6 is called Golgi tendon organ. And the last one is called joint receptors. These diagrams showing us different types of receptors. Several types, which are nerve ending, Pacinian corpuscle, Meissner corpuscle, and Merkel's tactile disc. As regards the free nerve ending, which is one type of non-encapsulated receptor, we will know that free nerve endings, they represent the most widely distributed nerve ending in the epidermis of the skin, especially in the hand and the feet. They also present in the mucous membrane of the visceral organs and cornea. It can be found also in serous membranes and in deep fascia. What about the pathway of the nerve ending or nerve fiber in this type? The nerve fiber loses its sheath and divide into many branches to terminate into knobs between the cell. Let's go to the diagram on the right side to see what happens. Which happen here, that free nerve endings start by myelinated nerve fiber. This myelin sheath is lost before penetration of the epidermis, then give branches which pass between the cells of the epidermis. They respond to noxious thermal and touch stimuli. What is the meaning of this function? That means that free nerve ending, its main function is to respond to pain, temperature, and touch stimuli which we can feel. The second type of non-encapsulated receptor is called peritracheal nerve endings. What is the other name of peritracheal nerve ending? It's called the plexus of Bonnet. Plexus of Bonnet, the site of this receptor is found around hair follicles. They are the nerve fiber which present around the connective tissue sheath of the hair follicle. What happens during the pathway of this nerve fiber? The nerve fiber loses its myelin sheath and branch to form basket-like arborization, which is present around the connective tissue sheath of the hair follicle. On the right side, we can see that the hair surrounded by connective tissue sheath, which is called the hair follicle. The nerve loses its myelin sheath, then turn around the connective tissue sheath to supply this nerve, connective tissue cover. What about the function of peritracheal nerve ending? The main function of plexus of Bonnet is to respond to displacement of hairs. The third one is non-encapsulated receptors is called Merkel's tactile disc. What about Merkel's tactile disc? Merkel's tactile disc presents as regards the site. It presents in the deep layers of the epidermis of the palmar surfaces of the hand and the plantar surfaces of the feet. Okay, what about the pathway of nerve fiber? The afferent nerve fiber was myelinated at first, but it loses its myelin sheath and Schwann cell, then expanded to disc-like structure, so it's called the Merkel's tactile disc, in relation to modified epidermal cell, which is called the Merkel cell. So it's called Merkel's tactile disc. Here we can see on the right side that the axon was myelinated, which is covered by blue color, then lose its myelin sheath and expand finally, making disc. These discs not penetrate, but become in relation to the cell which presents in the epidermal cells, which is called Merkel cell. So, what about the function of this Merkel's tactile disc? The main function is the mechanoreceptor, which respond to mechanical forces. As regard encapsulated receptor, as I have mentioned, that encapsulated receptors means that the receptors are covered by capsule. The first type in encapsulated receptor is called Pacinian corpuscle. You must know the site, structure, and function of each one. So let's go to know the site of Pacinian corpuscle. Pacinian corpuscle can be found in the skin, in the dermis of the skin. But it can be found also in deep structures inside our body, which is pancreas and periosteum and so on. What about the pathway and structure of this Pacinian corpuscle? As regard the shape, it is large ovoid structure. The afferent nerve fiber lose its myelin sheath after entering this corpuscle. Let's go to the diagram on the right side. We can see that this arrow point to the axon, which is covered by myelin. It is myelinated, entering the connective tissue capsule of the Pacinian corpuscle, but lose its myelin sheath inside, not outside the Pacinian corpuscle. Then after that, run axially to terminate at the end as club-like, as we can see in the photo, which is longitudinal section. Here on the right side, we can see two sections in the Pacinian corpuscle. The right side TS, which means transverse section, and the last one, the one which is on the left side, LS, that means longitudinal section. So I explain on the longitudinal section that the axon was myelinated at first, then enters the connective tissue capsule of the Pacinian corpuscle, then lose its myelin sheath inside the capsule, then the axon pass without cover, then terminally by the club-like at the end. After that, we can see that it is surrounded by numerous concentric layers of flattened modified Schwann cells. These give us the main shape or the characteristic shape of the Pacinian corpuscle, which is onion in appearance. Here we can see that the axon present in the center, on TS axon pointed in the center, then surrounded by multiple cells which are Schwann cells, but these Schwann cells are modified type, okay? On the LS appearance, we can see that the axon passed without myelin, okay? Then covered or surrounded by modified Schwann cells on around it. What about the function of Pacinian corpuscle? Its main function is mechanoreceptor also. It responds to deep pressure and vibration. Here, this is a test of vibration, which is responsible, Pacinian corpuscle responsible for this response. What about this photo? This is Pacinian corpuscle also, but stained by H&E stain section. This is H&E stain section for Pacinian corpuscle. The arrow point to the axon in the center, and all these structure which surround this axon are modified Schwann cell, and mainly this structure which is onion in shape covered by connective tissue capsule, as I have mentioned. This is the arrow point to axon. This is the axon. And these are modified Schwann cells which form concentric layers around. What about the second type of encapsulated receptor? The second type is called Meissner corpuscle. The site, you must know where can be found Meissner corpuscle. I can find Meissner corpuscle in the dermal papillae of the skin, of the palm of the hand, of the sole of the foot, and fingertips also. What about the structure? The structure is oval in shape, as I can see here on the right side. This diagram showing us a Meissner corpuscle, which is oval in shape. What happened for the afferent nerve fiber to enter? Afferent nerve fiber enter the base of the corpuscle at first, then branch, run in spiral course in between, transversely oriented flattened Schwann cell. Here we can see that the myelin sheath is lost before entering the capsule, then pass spirally inside, giving branch inside the capsule. Then I can see that there are multiple Schwann cells flattened surrounding the axon inside the capsule or inside the structure. What about the function? As I can see here, that this is a two-point tactile discrimination test. It responds to touch. Meissner corpuscle responds to touch, and two-point tactile discrimination test because it present in the dermal papillae of the skin, as I have mentioned. Here we can see that this arrow, which present on the right side, pink arrow, point to a Schwann cell. The blue arrow on the left side point to the axon, and the red arrow on the left side point to the connective tissue capsule, which covers the corpuscle. These are the photos of the Meissner corpuscle. Meissner corpuscle on the right side, this is stained by silver stain, and the left side is covered by. And the photo which we can see on the left side, this is Meissner corpuscle, which is stained by H&E stain. As regards the third type, which is encapsulated receptor subtypes, Ruffini corpuscle. What about Ruffini corpuscle? We must know the site of the Ruffini corpuscle. It presents in the deep dermis and subcutaneous tissue, especially in the plantar surface of the feet and the palmar surface of the hand. Here we can see the Ruffini corpuscle. This is a spindle-shaped structure which appear purple on the right side photo. This photo which present above, there are Meissner corpuscle, Merkel disc, and Ruffini corpuscle, and Pacinian corpuscle below. Ruffini corpuscle, let's see Ruffini corpuscle. They are found in the deep dermis, as I have mentioned, and present also in the subcutaneous tissue, where in the feet and the hand. What about microscopic structure? In shape, it is fusiform in shape. As regards the diameter, it is 1 millimeter by 0.1 millimeter. Each one has core inside, but this core containing collagen fibers, the nerve fibers lose its myelin sheath, okay, and the branches of this axon pass into flattened terminal between the collagen fiber. Here we can see the axon lose its myelin sheath, then enter the capsule and giving branches between the collagen fiber. What about the function? They are mechanoreceptors also. Okay. As regard the fourth type is called Krause's end bulb. What about Krause's end bulb? Krause's end bulb site, we can find this type in the conjunctiva, in the tongue, in the mouth, and external genitalia. This structure can be found in wet sources in our body. As regards the microscopic structure of Krause end bulb, as we can see in the photo here, Krause end bulb, thin connective tissue sheath covering it, thin connective tissue capsule, I can notice that soft gelatinous core present inside, and the myelinated nerve fiber lose its myelin sheath also, as the previous receptors lose its myelin sheath, then branch into network within the capsule. As regard the function, the same also as previous receptor, they are mechanoreceptors. Now we must know a very important comparison between the microscopic structure of two important receptors in our module. The first one, which is Bassanian, and the second one is Meissner. What about the differences between Meissner corpuscle and Bassanian corpuscle? Distribution, site, size, all of these are the main points which you will notice that you must talk about in comparison. As regards distribution, Meissner corpuscle can be found, as I have mentioned, in the skin of lips, fingers, external genitalia, non-hairy skin. But what about Bassanian corpuscle? They are widely can be found in subcutaneous tissue of the skin, of the palm of the hand, of the sole of the foot, in the fingers, and can be found in inner organs as pancreas. What about the site? The site, Meissner corpuscle, can be found in dermal papillae with its long axis which is perpendicular to the surface. As regards Bassanian, can be found more deep than Meissner, present in the dermis and hypodermis. What about the size? I can notice that Meissner corpuscle is smaller than Bassanian. So Meissner is small and Bassanian is large. As regards the other points of comparison between Bassanian and Meissner, the nerve fiber. What about nerve fiber in Meissner corpuscle? I have mentioned that it loses its myelin sheath before entering the corpuscle. As regards Bassanian corpuscle, so again, as regards the nerve fiber, Meissner loses its myelin sheath before entering the corpuscle, but the Bassanian corpuscle loses its myelin at the first node from VA, which is present inside the corpuscle. As regards modified Schwann cell, Meissner corpuscle containing Schwann cell which are arranged transversely as stacked lamellae between nerve fiber branches. But Bassanian corpuscle, the modified Schwann cell are arranged concentrically around the nerve fiber. What about presence of subcapsular space or not? Meissner corpuscle has no subcapsular space, but Bassanian corpuscle presents inside each subcapsular space, so subcapsular space absent from Meissner but present in Bassanian. What about the function? Meissner corpuscle is responsible for fine touch, 2-point discrimination test, but Bassanian corpuscle responsible for vibration and deep pressure sensation. Let's answer this question. One of the following is not a character of Meissner corpuscle. Number 1, it is an encapsulated nerve ending. It is present in the dermal papillae. The Schwann cells are arranged in 2 cores. The nerve fibers run spirally between the Schwann cell. Which one is not character of Meissner corpuscle? Let's see. Yes, the Schwann cells are arranged in 2 cores. Before this character is one of the characters of Bassanian corpuscle. Let's remember again, what about receptors? Receptors subdivided into 2 types, as I have mentioned before. The first one is called unencapsulated receptor. The second one is called encapsulated receptor. And we understood that unencapsulated receptor, not covered by capsule, subdivided into 3 types, which are free nerve ending, peritracheal nerve ending, and Merkel tactile disc. As regarding encapsulated receptor, there are several types: Meissner corpuscle, Bassanian corpuscle, Ruffini corpuscle, Krause's and bulb. And today, or now, we will discuss another types, which are muscle spindle, Golgi tendon organ, and joint receptors. What about muscle spindle? Muscle spindle is one of encapsulated receptor. The other name of this receptor is called neuromuscular spindle. Skeletal muscle, it present inside the skeletal muscle. It is known as the stretch receptor. What about the structure? Here we can see on the right side this photo. This is the section in the muscle, skeletal muscle fibers. In between skeletal muscle fiber, which is ordinary, between of them there are intrafusal muscle fibers which form muscle spindle. These fibers which are present in the center surrounded by connective tissue capsule, so it is capsulated receptor. What about the structure? It is fusiform in shape, as I can see here. It is capsulated receptor, yes, because it's covered by capsule, lie parallel to the muscle fibers. Each spindle contains 2 to 12 fibers inside, or intrafusal muscle fibers inside. 2 to 12 intrafusal muscle fibers. These muscle fibers, intrafusal muscle fibers, have very important criteria. They are smaller in diameter than extrafusal muscle fiber. They have indistinct striation. They are non-contractile central portion. That means the central part in these fibers non-contractile or cannot contract. What about this photo here? Here we can see muscle spindle or neuromuscular spindle on both sections, transverse section on the right side and longitudinal section on the left side. Transverse section on the right side, we can see that there is connective tissue capsule surrounding intrafusal muscle fibers, which are called nuclear bag and the nuclear chain. The first arrow points to nuclear bag, which is the large circle containing inside multiple nuclei, but nuclear chain, which is present below the nuclear bag, is smaller in size and containing one nucleus inside. What about the longitudinal section? Here we can see that these are strips or striations of a ordinary muscle, extrafusal muscle fibers, but between ordinary muscle, skeletal muscle fibers, there is fusiform capsule present here. Inside this capsule, there are two types of fibers, which are intrafusal muscle fibers. The first one, which is present above, is nuclear bag, and the second one, which is present below, is nuclear chain. Nuclear bag, we can notice here that the central part is bulged or dilated, containing multiple nuclei inside. And the second part, second type, is called the nuclear chain, containing multiple nuclei inside, which pass in row, and the central part or all the part of the nuclear chain is uniform in diameter. I can see here also that there are afferent and efferent nerve fiber which supply this intrafusal muscle fiber. The green one, which is called the primary afferent nerve fiber, surrounding the central part of intrafusal muscle fiber. As regard the orange fibers, which is called secondary afferent nerve fiber, present on both sides of primary afferent nerve fiber. And finally, the blue fibers, which are called efferent nerve fiber, supplying the contractile portion of the intrafusal muscle fiber. What about the type of intrafusal muscle fiber? Again, nuclear bag and the nuclear chain. These are the names of these intrafusal muscle fibers. As regard the shape of each one, nuclear bag, the shape is large and long, but nuclear chain is narrow and short. What about the number? The number of nuclear bag, because it is larger in size, so it is fewer in number. As regard nuclear chain, because it is short and narrow, it has numerous number. What about the nuclei? As I have noticed that there are many central nuclei inside nuclear bag, here we can see, and there are one row of nuclei present in the nuclear chain. So the shape in nuclear bag large and long, nuclear chain narrow and short. As regard the number, nuclear bag few in number, but nuclear chain numerous in number. As regard the number of nuclei and the site, the nuclear bag have many central nuclei, and the nuclear chain arranged in one row the nuclei inside. Innervation of muscle spindle. You must know that the muscle spindle, intrafusal muscle fiber which present inside, are supplied by nerve supply. These types of nerve supply are subdivided into two types: sensory or afferent nerve fiber, and the second type is called motor or efferent nerve fiber. Afferent nerve fiber, which is sensory, is called primary and secondary afferent nerve fiber. But these afferent nerve fibers have another names also. Primary afferent nerve fiber can be known by annulospiral, because on the diagram here we can see that primary afferent nerve fiber, which is called annulospiral, it is surrounded, making a spiral pathway around the central part of nuclear bag and the nuclear chain. As regard the second type, which is called the secondary afferent nerve fiber, is called the flower spray. Flower spray present on both sides of the primary afferent nerve fiber or annulospiral nerve fiber, and present on both sides of non-contractile portion. As regard motor or efferent, nerve fiber, I can see that they are present on both sides of the nuclear bag and the nuclear chain, supplying the contractile portion of these fibers. What about the function of muscle spindle? They are stretch receptors of skeletal muscle. They are stimulated by the degree of the muscle stretch. They give information for position and the movement of the muscle. What about stimulation? They are stimulated by what? They are stimulated by stretch, either active stretch or passive stretch. Active stretch, that means contraction of contractile part of intrafusal muscle fiber. That means when you contract the contractile part or central part of intrafusal muscle fiber, that means you made active stretch. What about passive stretch? How can be done? It can be done by pull on the tendon, as in knee jerk test. As regards the second type of encapsulated receptor, is called the Golgi tendon organ. Golgi tendon organ is also stretch receptor. But what about the difference between Golgi tendon organ and the muscle spindle? Here we can see that the Golgi tendon organ, the site present at different site from the muscle spindle. Muscle spindle was found here, where was found between extrafusal muscle fiber of the muscle itself. But as regard the Golgi tendon organ, it can be found at the junction of the muscle with the tendon. Here we can see on these photos or diagrams that contact between the muscle and the tendon. Here we can see that there is Golgi tendon organ. What about the microscopic structure of Golgi tendon organ? It is encapsulated spindle. What about encapsulated? What is the meaning? It means that it's covered by capsule. So it is encapsulated spindle, which is shaped structure like spindle. Differ from muscle spindle in what? In its site. So again, as I have mentioned, that there are differences between muscle spindle and Golgi tendon organ. In what? In the site. Because I have known that the muscle spindle present in the muscle itself between extrafusal muscle fiber. But Golgi tendon organ, where can be found? It can be found at the junction between the muscle and the tendon. Also, it differs from the muscle spindle that it contains collagenous fibers, because muscle spindle have no collagenous fibers inside. The third difference between that Golgi tendon organ lacks efferent innervation, but you know that the muscle spindle have efferent nerve fiber which innervate it. What about the function? They are stretch receptors also, but give the information to central nervous system as regard tension in the muscle. What about effectors? The second part in our lecture today is called effector. What about effector? Here we can see that the receptor present at the end of sensory nerve fiber, and in contrast of this, I can notice that the effector present at the end of motor nerve fiber. What about ILOs? By the end of this lecture, you will be able to describe the structure and the correlated function of motor end plate. Receptors, again, let's to remember that they are subdivided into two types. These two types are in unencapsulated and encapsulated receptor. Unencapsulated, which not covered by capsule, subdivided into free nerve ending, peritracheal nerve ending, and Merkel tactile disc. As regard encapsulated receptor, which is covered by capsule, subdivided into Meissner corpuscle, Pacinian corpuscle, Ruffini corpuscle, Krause end bulb, and muscle spindle and Golgi tendon organ and joint receptor. This is just a revision on the receptors. What about effectors? Effector present, as I have mentioned, at the end of motor nerve fiber. Here we can see that this is transverse section in spinal cord. Spinal cord containing inside gray matter which take the shape of H shape. This H shape has anterior horn present anteriorly and posterior horn which present posteriorly. From anterior horn arise motor nerve fiber which at the end of this they are found, effectors. But the posterior horn to which afferent nerve fiber or sensory fiber pass to enter at this place at the end of this sensory nerve fiber containing receptor at the end. Let's talk about effectors. These are termination of the peripheral nerve fiber. Which peripheral nerve fiber, sensory or motor? No, motor. Motor nerve fiber in the effector organ. They are classified into two types, somatic effector and visceral effector. What about somatic effector? Somatic effector supplying muscle spindle. Skeletal muscle fibers. In this case, it can be named by motor end plate. As regards the second type, which is called the visceral effector, supplying what? Supplying autonomic organs, which supplied by autonomic nervous system, such as cardiac muscle, smooth muscle, and glands. As regard motor end plate, what are the important points you must know about? Number one, you should know that it can be stained by another stain, not H&E. It can be stained by gold impregnation technique. Okay, it is the site of axon termination from AHC, that means anterior horn cell, which is present in the anterior horn of spinal cord, as I have mentioned. So it is a site of axon termination from anterior horn cell on the skeletal muscle fiber. Each skeletal muscle fiber is supplied by one motor axon. So there is the important expression. You must know what is the meaning of motor unit. Motor unit equal anterior horn cell, one motor neuron, plus one motor axon with its branches, plus skeletal muscle fibers that innervate. Here we can see on the right side that these are skeletal muscle fibers, and this black structure which pass to branch and come in contact with the muscle fiber, these are the axons. Then axon, when becoming contact with the muscle fiber, is called in this case motor end plate. What about motor unit? Motor unit, here we can see that the anterior horn cell, which is present in the anterior horn of the spinal cord, gray matter spinal cord, there are two axons arise from here. The first one, which is red in color, and the second one, which is purple in color. The red one is called motor unit 2 because it is giving branches, and these branches become in contact with the skeletal muscle fibers. Okay, So axon terminate and the neuromuscular junction here, this is called motor unit 2. As regard motor unit 1, this nerve fiber which arise from the anterior horn cell, it is purple in color, arise from anterior horn cell passing through the anterior division, then giving branches, then becoming contact with multiple muscle fibers. So all the muscle fibers which are supplied by the same branches of the same axon which arise from anterior horn cell are called motor unit. What about the LM, light microscopic feature of this structure, which is called the motor end plate? Motor end plate is a site of contact between axon and the muscle. It's called motor end plate. Yes, I have known that. Axon, what about the pathway? The axon was myelinated at first. It loses its myelin sheath. Schwann cell remain to cover the axon, then branches into many terminal. Each forms depression on the sarcolemma, which is called troof. Troof space between axon terminal and sarcoplasm is called primary synaptic cleft. Here we can hear new expression. What is the meaning of exoplasm, sarcoplasm? Okay, let's talk about sarcolemma. Sarcolemma means cell membrane of muscle fiber. What about sarcoplasm? That means cytoplasm of muscle fiber. Okay? Here we can see the light microscopic feature of the motor end plate. On the right side we can see that this is basal lamina. Okay? I can see here that the pale area here, this is the nerve terminal, and the dark area below, this is the muscle terminal or the muscle part which become in contact with the nerve terminal. Inside the pale area we can find mitochondria of the axon. These are mitochondria, and these rounded structures, they are called presynaptic vesicles. Here we can see that there is a space between the axon terminal and the muscle fiber, which is called the primary synaptic cleft. Below, I can see the dark area containing mitochondria, but these are the mitochondria of the muscle cell, and the muscle surface or cell membrane of the muscle fiber is folded into junctional fold of the cell membrane. Okay. What about the EM? What can be found in the electromicroscopic structure of the motor end plate? Here on the right side in this diagram we can see that the axon was covered by myelin sheath, which is orange in color. This axon loses its myelin sheath and making a wide area which become in contact with the muscle fiber. Okay, inside the cytoplasm of the axon I can see that there are mitochondria and presynaptic vesicles, which are rounded structure present at the cell cytoplasm or the axon cytoplasm. There is a space which is present between the axon and the muscle, which is called cleft. Okay, and I can see that the muscle side is folded up and down. These folds are called junctional fold. So sarcolemma or cell membrane of the muscle fibers have many enfoldings. These are secondary synaptic cleft or junctional fold. What about sarcoplasm? That means cytoplasm of muscle fibers. It contains inside several nuclei, numerous mitochondria, and containing acetylcholine receptors. What about exoplasm? That means cytoplasm of the axon, containing numerous synaptic vesicles and mitochondria. What about the cleft? The cleft between, containing choline esterase enzyme. Function of the motor end plate. Here we can see that arrival of nerve impulse in the axon cause depolarization of axolemma or cell membrane of the axon, which leads to acetylcholine release from presynaptic vesicles. to enter synaptic cleft between the muscle and nerve. After that, what happens? It binds to its receptor, which is present on the postsynaptic membrane, which lead to depolarization of sarcolemma. After that, muscle contraction occur. What about this quiz? How many skeletal muscle fibers comprise a motor unit? One or many, or any number from 1 to many depending on the muscle? The correct answer is many. What about this complete question? Nuclear bags are space, while nuclear chain fibers are space. Nuclear bag fibers are large and long. Excellent. While nuclear chain fibers are short and narrow. Yes. The second question, nuclei of nuclear chain fibers are space, while nuclei of nuclear bag fibers are space. As regard nuclei of the nuclear chain fibers are arranged in one row, like the diagram here, while nuclei of the nuclear bag fibers are numerous and central. The sense organ most closely associated with stretch reflex, such as knee jerk, is what? Meissner corpuscle, Pacinian corpuscle, muscle spindle, Golgi tendon organ, or free nerve endings? What is the correct answer? Excellent. Golgi tendon organ. Thank you so much, and thank you for the listening. Good luck.